When it comes to understanding the relatedness of certain strains, it can be useful to try and figure out the presence of any differences in the length of their chromosomes. In some eukaryotes, like Cryptococcus, large translocations or recombination events can accumulate over the course of generations, creating what we call chromosome length polymorphisms. Visualizing the length of really enormous pieces of DNA like chromosomes is impossible using normal gel electrophoresis. So we use something called a pulsed field gel electrophoresis, or PFGE. We use a system of clamped, homogeneous electric fields, or SHEF, to slowly wiggle large chunks of chromosomal DNA through a specialized high-resolution agarose matrix. We'll talk through exactly how the system works a little bit more together in class, but for now, just think of it as a huge agarose gel that requires a little bit of extra legwork to set up. The most important part of this process is ensuring that our protoplasts stay in good shape, keeping the integrity of the nucleus and preventing the chromatin from degrading, which will keep us from seeing nice, clean bands. Let's get cracking. First off, we're going to embed our clean, freshly digested protoplasts into some agarose. We're going to give them a quick spin down to pellet them at the bottom of our epindorfs, decant our supernatant, and very carefully rinse the pellet with some SCE buffer. Just like when we prepared our protoplasts, the sorbitol acts as an osmotic support to ensure that our delicate protoplast suspension is kept isotonic. We want to avoid damaging the protoplasts as much as possible at this point, so we're not going to resuspend the cells, just washing the top and tipping it off a couple of times. The next step is extremely time sensitive. We're going to resuspend our protoplast pellet in a little bit of low melt agarose that we've kept at 37 degrees Celsius, just slightly above the temperature it starts to set at. We keep the temperature this low to avoid adding stress to our protoplasts, but it can start to set quite rapidly. Note that when we pipette up and down to mix our protoplasts into our agarose suspension, we're using a truncated tip. This is so we can limit the mechanical damage to the protoplasts at this stage, as they can shear and burst when they're taken up through the very small aperture of the tip. Now that we've embedded our protoplasts into agarose, we're going to make some small plugs that we can load into our final gel. Using a yellow truncated tip, we're going to very carefully pipette the protoplast suspension into these plug molds, so we can have inserts of consistent thickness across all of the strains we're preparing. The agarose will set very quickly, so it's vital that you're careful and act fast. Once our plugs are set, we can remove the backing strip and use the small tool to gently ease them out of the mold. Now they're free, we're going to place them into an overlay solution. We'll talk a little bit more about the solution in class together, but for now I'll just say that this is a buffer that contains a gentle detergent that will lyse the protoplasts in the plugs, and some proteinase K, which will non-specifically chew up proteins in the protoplast. Most importantly, this proteinase will break apart the histones in the fungal nucleus, unpackaging the DNA and letting it move freely through the final gel. We're going to incubate our mix at 50 degrees Celsius for 24 hours to let these reactions run their course, and then remove the overlay solution and gently rinse and store the plugs in 0.5 molar EDTA. Once we're ready to load our gel, we can gently rinse our embedded chromosomes with some TBE buffer and pick out the most even looking plug that we made. We only need one decent one. Drying it gently by blotting it onto some lab tissue, we're going to load it onto our PFGE well former and take a scalpel and carefully trim it down to size so that it fits. Just like with a normal gel, we still need some size standards, both to compare our chromosome lengths to and as a control to make sure our gel has run correctly. We use a thin slice of some agarose embedded with chromosomes taken from Saccharomyces cerevisiae, a very well characterized model organism with very well established chromosome lengths. Once we've let our plugs dry on the teeth of the well former, we can load it onto our chef gel casting stand and gently pour some low melt, high grade agarose around it. It's important to be gentle here, as we don't want our chromosome plugs to float away. Let it set, and now our gel is ready to be loaded into a chef apparatus to have a pulsed field run through it.